Good afternoon, everyone, and thank you for joining us on this exciting presentation from the Australian Museum. My name is Megan Warwick, and I'm going to be the moderator for this webinar. So, although we meet from different parts of the country, I would like to acknowledge the Gadigal people of Eora Nation, the traditional custodians, the land the museum is on, and pay my respects to elders past, present, and emerging. So today we are delighted to have Professor Christopher Helgen, the Australian Museum's Chief Scientist and Director of the Australian Museum Research Institute, or AMRI, and our paleontologist, Dr. Patrick Smith, who will be providing an update on the paleontology projects currently being undertaken at the Australian Museum. So I just wanted to run through a few housekeeping things first. This webinar is being recorded and it will be available to share on the AM website. At the end of the presentation, there'll be a 10-minute Q&A session, so please use the Q&A function to ask questions at any time. So I'd like to give a very warm welcome to all of our donors, our partners, and our members. Thank you so much for joining us. We are also joined by the directors of the Australian Museum Foundation and the Australian Museum Trust. So to learn more about AMRI and our paleontology department, I would like to introduce you first to Professor Christopher Helgen, Chief Scientist and Director of AMRI. Welcome, Chris. So Chris has been with us. Chris has been with us for just over a year now, and he's responsible for more than 100 staff, including our research scientists, collection scientists, collection officers, and more than 130 associates, fellows, and students who research and explore today's major challenges. So Chris was Professor of Biological Sciences at the University of Adelaide, and he's focused his research primarily on fieldwork with living animals and research in museum collections to document the richness of life, explore global change, and contribute to important problems in biomedicine. So originally from Minnesota, he served as curator in charge of mammals at the Smithsonian's National Museum of Natural History in Washington, D.C. So Chris has been visiting the Australian Museum for over 20 years since his student days with Professor Tim Flannery. So welcome to Chris.
Thank you so much for that introduction, Megan, and it's a pleasure to be here with all of you today and to introduce our program today about paleontology at the Australian Museum. Now, it's my enormous honor as the Chief Scientist at the Australian Museum, as Megan said, to oversee tremendous programs in science at the museum that involve our researchers and our phenomenal collections. And so, as you've heard, we have more than 100 people that work largely behind the scenes at the museum, working on scientific questions and problems, as well as looking after our world-class collections. Now, when you think about coming to the museum, usually people are thinking about the things that you can see front of house, the exhibitions, the galleries, the educational aspects of the museum, and these are core to who we are. But a very small percentage of all the treasures, the specimens, the biological and geological and cultural materials that we hold are on display. Most of them are behind the scenes in the collections. And we have many different kinds of scientists that work on that material. What are we talking about? Studies of life sciences, where we use incredible collections of everything from mammals and birds, reptiles, spiders, insects, marine invertebrates, basically samples and specimens taken from the last hundreds of years that have come into the museum, are looked after carefully behind the scenes, and form the basis for understanding and piecing together the aspects of our natural world. How many species live here in Australia? How many have lived here in the past? How do they come together to become the ecosystems that make Australian natural environments tick? Where is our place in nature, the cultural richness of Australia and other parts of the world? And how does the planet and its life, its environments and its climates and human culture all work together? These are questions that we love to study behind the scenes and that our collections give us a unique way to address. Now, one of the collections we're going to talk about today, and one of the research aspects of the museum, is paleontology. And this is, of course, the study of fossils. This is always a crowd-pleasing aspect. It is so important that in a museum like ours, where we have large collections of fossils assembled, not just from decades and centuries of work by our scientists and our technical officers, but materials that actually sample millions, tens of millions, hundreds of millions, even billions of years of geological history that give us a window into life on Earth and how it has changed through time. So this is what you're going to learn about today a little bit, the way that we approach the study of past life by detective work, scientific research on fossils in our Australian museum collections. And you're going to hear about a number of projects. Now, it gives me great pleasure to introduce one particular researcher on our staff, Dr. Pat Smith. Now, Patrick Smith is an invertebrate paleontologist, which means that he studies fossil animals that have no backbones. And in particular, Pat did his PhD on trilobites, which are a group of marine arthropods, segmented animals with paired appendages. These are one of the most successful groups of animals in the history of life for a long period of time in the fossil record, long before dinosaurs, until their eventual extinction. But they're an important group to understand patterns of life and evolution on Earth. And this is Pat's great expertise within paleontology. Pat is also going to explain and tell us about projects that involve other paleontologists at the museum and the kinds of work that they're doing, the projects that excite them. And doing so, he's going to paint an overall picture for the kinds of work that we do studying fossils in the museum. Now, Pat is, in addition to being a tremendous researcher who's publishing papers, he's also has a role as technical officer looking after these incredible collections in the fossil part of the museum, in the paleontology collections. And so you can imagine what that would be like for someone like Pat. For all of us, it's something like a kid in a lolly shop. The difference here is that even though you can see on the screen he looks very young, this is no kid. This is a very highly experienced scientist, and it's no lolly shop. We do not eat the fossils, but we do look and study them in many, many, many other ways. So it's a great pleasure to introduce Pat. I'm going to turn it over to him to tell us about the work that is done behind the scenes, studying paleontology, the history of life, using the Australian Museum collection. Over to you, Pat.
So, g'day. Thanks very much, Chris. Yes, as Chris introduced, my name is Patrick Smith, and I work at the Australian Museum in the paleontology collection. I particularly work with the collections, databasing our collections. We have over 150,000 objects, which we need to database so that we have an idea of where they are stored as well as where they come from. And these assist researchers in their own research. And so today I'll be talking to you about some of the projects that we've been working on, particularly the cutting edge of our projects, so that you'll have an idea of some of the research the Australian Museum has been doing. So some of our new discoveries involve organisms that aren't dinosaurs. And in fact, one of the things we commonly get asked at the Australian Museum is aren't you as paleontologists just always studying dinosaurs? And that's just not the case. So one of the messages I want to have as a take home for those listening is that paleontology is more than dinosaurs. What paleontology really is understanding change over time. So as paleontologists, we study geological time. So I've placed a geological time scale here on the slide that you can see to your left. And as you can see, there's a large number of periods that have different names. And those different periods with different names, some of them you might recognize, like Jurassic, from things like Jurassic Park, of course. But others are maybe a bit more unfamiliar. Some of these geological periods are placed numbers next to them. Those represent millions of years. So for example, the Jurassic period started about 201 million years ago. So if we look at our geological timescale, if we think about things like dinosaurs, for example, we see that they appear relatively late in the stage of Earth's history, particularly the stages of life that we have. So if we look, for example, at the dinosaurs, I've placed a marker there, they appear at the last gasp of the Triassic period, so only a little less than 201 million years ago. If we look at, for example, plants, they are much further back. They're sometime between 358 to 419 million years ago they first appear, and the first corals in the fossil record appear at about 510 million years ago. So you can see life on Earth has had a very long history, even before the dinosaurs. So studying life, we can understand things like change, for example, change over evolutionary and geological timescales, like, for example, our own relatives, so looking at human ancestors, for example. But you can also look at change on smaller timescales. I've known paleontologists who are collaborators of mine who work on climate change, for example, so human-induced climate change, looking at changes in shells, for example, in Sydney Harbour over the pre- and post-European settlement periods. And we can see, for example, changes in faunas over that time. So paleontology is all about change. So the three projects I'll highlight to you, I'll be going further and further back in geological time as I explain them. So the first one is on a site we call McGraths Flat. This is a site that was found by one of our research associates at the museum. His name is Robert Beattie. He's a school teacher. And he was looking for fossil insects. He went to a site just north of Gulgong. We can't actually tell you where the site is. It's a secret. But we went to a site north of Gulgong, and he got Dr. Matthew McCurry, our curator of paleontology, and Tara Djokovic, who's one of our researchers involved in this project. And they've been researching and understanding what this site's importance is to us in terms of change over time. So we'll have a look here. You can see that it's Middle Miocene in age, this site. So it's about 15 million years old. So it's a relative baby if you'd like to think of it in terms of its geological age. And this Miocene site, when Robert first came to it, he found a bunch of plants. Now, he was looking for fossil insects, but he stumbled across the plants first. And it was quite a large diversity of plants that he managed to collect from the site, as well as later trips that we've done with him. And they include a variety of plants from different environments. So for the example, on the left, on the top three images here, these are rainforest-style plants. For example, the first one on the left there is Lygodum, which is the climbing fern. But also we have plants with drip tips and also plants with serrated edges, all of which suggest a tropical climate, so something with high levels of precipitation. On the lower part of that left-hand side of the image, we have dryland species that also occur in amongst this fauna. So things like, for example, the she oaks, as well as myrtles. What you're seeing on the left-hand side in the lower image there is actually a bunch of flowers, fossil flowers that are preserved. You can see the anthers, which are the pollen-bearing elements of the plant, sitting in there as those yellow sort of tufts. Now, the fossils, whilst they look very beautiful at a macroscopic scale, we can also zoom in and see some highly intricate details on those plant fossils. So, for example, over in image H here, so that there is a stomate. So it's one of the cells that guards the leaf of a plant. And that guard cell allows the gas exchange that you get with oxygen and carbon dioxide in and out of a leaf. It's quite remarkable, really, because these have not previously been reported from plants of this age in Australia. We also found not just the plants themselves, but also pollen. So, for example, we find pollen of dryland species, which is what you see on the left-hand side here in those four images. And these are pollen which we get as internal and external molds. The actual pollen has dissolved away, and all we're left with is basically the internal and external components of what the pollen would have looked like. However, despite the fact that we just have sort of a mold of the pollen, we're actually still able to identify it down to a species level, and that gave us that Middle Miocene age, that 15 million year old mark. Interestingly, these pollens would tell us that this site is a unique site because previously people weren't able to extract pollen from rocks of this type. This is called a ferrocrete. And previously people had to dissolve rocks out in acid, typically carbonates, so rocks that are full of calcium carbonate. And they would do that to reveal the pollen and use that to identify the ages of the fossil sites. However, what we found with this site is that we can actually just image the rock directly under a scanning electron microscope. Some of the other pollen that we have, for example, the M&N specimens that you see there, those hexagonal-shaped bits of pollen, are in fact from a Notrophagus. So this is a southern beech tree. It's one of the trees that's still around with us today, though it doesn't live at the site, as I'll explain a little bit later. Of course, we don't just have plants from the site, we also have animals, and Robert was ecstatic to find that we had fossil insects there, which is what he was initially looking for. The one on the right-hand side, there's this large wasp, is indeed what you think it is. It is a large wasp. It's a purgid wasp. It's a primitive sort of wasp. We also see that it has its wings preserved in immaculate detail, which has enabled us to identify it as a new species. On the left-hand side, we see a fossil feather. So feathers, for example, this would have come from a bird. And not only is the feather preserved as individual barbs that you can see on the feathers, but if we zoom in on those barbs using a scanning electron microscope, just like with the pollen, we can see small, in this case, subcellular structures. These sort of rod-like elements that you're seeing are in fact what we call melanosomes, which are the bodies that sit inside cells or produced by cells that give pigment and color to organisms. And what that tells us, based on the shape of those melanosomes, is that this bird was iridescent or black in color. And based on its size, we can tell you it was a relatively small bird too. So this enables us to basically, for the first time, identify even what, for example, the color of these birds were in the environment that they were living in. So extraordinary detail is preserved. We don't, of course, just have terrestrial organisms like those, for example, the plants or the animals that I was just showing you, but we also have aquatic organisms, as you might expect in a fossil deposit. Fossil deposits generally have aquatic organisms because they're formed generally in lakes or rivers, or sometimes in oceans. And so we also find fish, for example, in this deposit. You can see on the left-hand side, this is a fish. You can see his eye preserved there, along with his long spinal column running down his back. And inside the fish, you see a large blob-like structure. This is, in fact, its preserved gut. And inside that gut, we have these long filament-like elements. Now, those elements, we actually managed to match them with some of the other fossils we were finding in the deposit. So on the right-hand side, you'll see that this is what we call a phantom midge. It's a larvae from a phantom midge, a bit like a mosquito, if you want to think of it that way. They're close cousins. And here's a modern phantom midge on the far right there for comparison. You can see in both cases they have these long raptorial appendages at the front. These are actually modified antennae that they use for feeding. They use it like a bit like a basket, and they sort of feed on some of the algae that's floating in the water. And so what this fossil fish is doing is actually showing us a really nice example of almost a preserved ecological food chain. What we have here is we know the algae were being fed on by these phantom midges, and we know that the fish were feeding on the phantom midges. And so we have a nice preserved, almost single-line food chain there in that one fossil. The same fossil also produced some interesting results as well for looking at melanosomes. So it also has melanosomes like that feather. However, in this case, they're preserved inside the eye. And in fact, we find that they're spherical in shape in this situation. And these spherical-shaped melanosomes tell us it was probably likely somewhat reflective, a bit like some modern fish that have semi-reflective eyes. And so we can actually start to guess at the color of the fish. And this can help us understand, again, the ecology of what's going on between these organisms. Now, one of the surprising finds at the site also was fossil spiders. So this is a fossil spider that you can see here. Now, fossil spiders are extremely rare, and that's because spiders, like insects, are made of material that breaks down after the animal dies, and so they generally don't preserve. They also don't generally live anywhere near water unless they're water-dwelling spiders, but even in that situation, they're very uncommon. And so we see here that this spider is preserved not only in its entirety nearly, but also it's preserved down to, again, very fine details that are, in this case, even visible under just a hand lens. You can see the individual hairs that sat on the spider's leg. Now, it's great that we have this spider, and it's a new species as well, and it's great to have new species coming out of the ground, but what do they really tell us about fossils and change over time? I mean, if paleontology is about that, new species don't tell us much except that there are species that have gone extinct. So what this tells us, this site, is that, as you can see, there's a lovely reconstruction that's been done here based on the insects and plants that we've been finding. It was more of a rainforest-style environment, and in fact, using the plants themselves, and particularly the leaf shapes, we can tell you that precipitation was much higher than it was today. If we look on the right-hand side, this is a photo from the site as it is today, it's much more of a grassland, and in some places a bit of a dry sclerophyll forest. But if you compare that to the reconstruction on the left, you can see that this is a highly productive ecosystem that these fossils came from, much closer to what we would see in, say, a tropical rainforest or subtropical rainforest today in Australia. Now, if we plot out the ranges of some of the groups of organisms that belong from the site, you can see the site there denoted by the star on the map. You can see that a lot of them actually seem to have moved away from the site. They didn't adapt or evolve to the changing conditions as Australia was drying out over the 15 million years up until now. But in fact, they migrated towards either the edges of the continent, or they moved into other continents that were also much more had much higher precipitation. For example, the Notrophagus you find in New Zealand as well as New Guinea, for example. The master termis is the master termites that you find in the tropics today, and the caddisfly larvae, tube-dwelling caddisfly larvae, as you can see, hug the coastline of Australia, again because of the precipitation. So because of a drop in precipitation over time, these organisms have had to reduce where they've lived in Australia. And so this tells us a lot about how organisms adapt and change to change. They don't seem to adapt to their local environment in a lot of circumstances, but just move to where environmental pressures are more suitable for them. The second project I'll be talking about goes back to the time of the dinosaurs. So we'll be looking at one of our PhD students, Lachlan Hart, who's been working with Dr. Matthew McCurry, looking at Mesozoic croc-like amphibians. The Mesozoic is the period in time in which the dinosaurs were around. And what we see here, Mesozoic amphibians have been known for Australia for a long time, as well as from elsewhere around the world in much older rocks. But the ones from Australia are particularly interesting because during the Mesozoic, these guys seem to have declined elsewhere around the world. Now, we've known of these species, in particularly around Sydney, for a long time. This is a species here that you see in the centre of the image. What you're actually looking at is a skull with the red arrow pointing to the eye. You can sort of orient yourself after you see the eye and where the jaw is. This is actually a nicely preserved skull of one of these animals. Now, these temnospondyle amphibians are more closely related to things like frogs or newts and salamanders than they are to crocodiles, despite looking like them. And the first one I'll be talking about comes from an interesting period in time known as the Triassic period, which occurs just after a period known as the Permian. The Permian is interesting because it occurs just after a mass extinction event. Now, the rocks around Sydney in particular preserve this remarkably well. And if you go to places like Ostermere Beach, which is in the photograph on the left there, you can actually see the boundary between the Permian and Triassic periods, and it's quite distinct. And it's because of this extinction event. The extinction event wiping out 50% of marine life, as well as wiping out potentially estimates up to 90% of terrestrial life at the time. As I said before, these temnospondyle, these crocodile-like amphibians, were really common around the world in the Permian, and even earlier than that. However, we find that after this in the Triassic and Mesozoic, they seem to drop off in terms of their numbers. And in fact, they seem to almost be replaced by crocodile-like organisms that seem to start to dominate. There's an example of one of these crocodile-like organisms and their relatives up the top here. This one's from Tasmania. You can see there's parts of its skull, and you can see on the right there a nice reconstruction. And in fact, when I say crocodile, these animals didn't act like modern crocodiles, but probably much more like terrestrial mammals did. So the first site is one near Kingcumber, which is sort of near Terrigal on the central coast. And looking at the site, this is an outcrop nearby. You can see that there's very fine layers of shale and sandstone, which we think likely were deposited in a lake. To support this idea, it comes from the fact we also have ferns preserved. There's no large trees preserved at this site, and it's likely that just after this mass extinction event that killed off so much life, the plants had to recover just like animals did. And so only ferns were deposited in this lake, as they were the only things living next to it. Inside the lakes itself, it seems like they had fish and large sharks. There's an example down the bottom on the left there of a xenocanth shark. There's a large shark that's found in slightly older rocks in the Sydney Basin. But we've found similar ones in similar aged rocks nearby. And the idea is that these were deposited in a large, what we call braided river system, a bit like the modern Brahmaputra in India. If you look at the modern Brahmaputra, which I have in the centre of the image at the bottom there, you can see that there's these large rivers that sort of form kilometres worth of deposits, and they're very unsupported banks, which tend to erode very easily. Now, the site itself has produced a remarkable specimen, and Lachlan is currently working away at describing this as a new species. So this is one of the amphibians. It's affectionately known as the King-Cumber amphibian. It was found in the early 90s in a retaining wall of all places. Somebody was building a retaining wall and they just happened to saw out a block of stone that had this amphibian in it. After careful preparation by Lachlan, he actually has revealed parts of the skull that you can see in the top of the center image there, and also around the sides of the body of the animal. In fact, we think it potentially has skin preserved. Now you can see in this specimen, it has the near complete body, which would make it the second only complete specimen of a temnospondyl known from the Sydney Basin. You can see from the size of the animal in the reconstruction on the right, it was quite a large animal. It was likely the top predator in its environment at the time. It's quite a remarkable specimen. Moving slightly more forward in time, so we're getting younger, we see the second site that Lachlan was focusing on is another one north of Golgong. This is a site called Taubrigga. And at Taubrigga, it's a different sort of environment to the first environment. This one, being slightly younger, is actually near a volcanic eruption site. It was likely a lake that was inundated either several times or in one large event, although it's more likely several times, by volcanic ash that was coming out of these volcanoes and suffocating what was ever was living in the water or around in the environment at the time, including very nicely preserved fish as well as plants. But of course, Lachlan wasn't interested in those. He was interested in this fascinating tooth. This is a single tooth, and it's one of only a single tooth that's been found at the site. And what he did was he managed to get CT scans of this tooth. Now, you might think that why would you bother CT scanning a tooth? But the internal structures of teeth can tell you a lot about the origins of some of these animals. And before we'd CT scan the tooth, we didn't know what the tooth would have belonged to. It could have belonged to any number of organisms. A dinosaur, could have belonged to a large fish. However, Lachlan managed to demonstrate that inside the tooth were in fact a labyrinth-style passages that connected to the central canal. If we look, for example, in the center at the top there, you can actually see a nice reconstruction of these as we move through slices of the teeth at different levels. And these only occur in several particular groups of organisms, and the one group of organisms which it most likely belongs to, because there's nothing else in the environment that it could have belonged to, is a temnospondyl amphibian. And so we can demonstrate that this is a Jurassic temnospondyl amphibian, and that's unusual. Not in Australia, because we've known of Jurassic temnospondyl amphibians in Australia, but elsewhere in the world they seem to have gone extinct in that earlier Triassic period. Now, we know from deposits elsewhere in Australia that dinosaurs were wandering around at this time, and so it's likely that, like the other temnospondyl from the Triassic, it was also the top predator at its time and probably was feeding on fish, sharks, and as well as small dinosaurs that got nearby. This reconstruction you can see on the right here has a small fluffy dinosaur being eaten by an amphibian, which is something that we don't typically consider when thinking about fossil amphibians or newts, that they were feeding on things like dinosaurs, for example. We tend to think of dinosaurs as the top predators. So, okay, it's nice to know that we have two new species of a temnospondyl amphibian, but what does this tell us about change? Well, temnospondyl amphibians, as I said, seem to have done really well early on in the history of life. However, they seem to have gone extinct elsewhere into the Mesozoic, so into the time of the dinosaurs, except in Australia. Why did they persist here? Well, it seems likely that it has to do with both environmental and geographic factors. Like with the McGraths Flat site, the previous project, as environments change, organisms seem to restrict their ranges to preferable habitats. Now, one of the unusual things about Australia at the time is that the continent of Australia, which has moved, is actually down near where Antarctica was during the Mesozoic. And we have evidence for the fact that it was exceptionally cold at the time, coming from things like what are called drop stones. This is what you're seeing in the top on the right there. These are actually stones. This is a photo I took in New Zealand, sitting on top of a glacier. And as these glaciers float out onto lakes and rivers and into the ocean, they drop rocks. And these rocks get preserved. That's what you're seeing in the center of the image there. Along with that, we also have a pseudomorph, or a type of crystal called a glendonite. This is a crystal that's modified from another mineral called ikite, and that tends to form only in sub-zero conditions. And again, these are preserved in the Mesozoic of Australia. So Australia seems to have been exceptionally cold, and that may have played into the fact that these guys could survive, these temnospondyls could survive, where they couldn't elsewhere in the world because Australia was so exceptionally cold. It may have also been that Australia, being so remote and out in that limb of Gondwana that you see there, those continents all merged together, meant that the geographic barriers isolated and stopped other organisms that were taking the same role as these temnospondyl amphibians. And so again, we see that change drives where organisms live, and that as organisms become more and more restricted, they potentially eventually can become extinct. The third project I'll be talking about is much older than the other two projects. It's a project that I'm working on myself. It's on Australian Ordovician trilobites. So it started off really with actually these two specimens here. The first was only seen in a Kodachrome slide. You can see it's a photograph. We have the specimen sitting at the museum. And it was found by this fellow whilst they're making a documentary in 1997 in outback Australia. It's a nautiloid that's a straight-shelled animal that looks a bit like a squid. You can see a modern nautilus there on the side just for comparison. The shell types, they're sometimes quite different. As well as this large trilobite here, which was found in the collections of Geoscience Australia. This is the tail of a trilobite, or what's called the pygidium. So I went looking for more of this material because those two large specimens surprised me because nobody had reported species of those sizes from this period in time anywhere in Australia before. And so we went looking at where these specimens were found. They were both found in the same spot near Alice Springs in what's known as the Amadeus Basin. And you can see there's a photo of myself as well as one of our volunteers, Patrick Nelson there. We were wandering across outcrop looking for more material as well as driving across it. And at this time, Australia was a very different place. It wasn't the continent that we know today. Mostly what was land at the time was in Western Australia. And what was in the ocean or in the sea was in the Far East, towards what we now think of as the eastern states of Australia. And in fact, the east coastline didn't exist. Most of what we know of the eastern coastline didn't exist at that time. So what we were looking at is rocks that were formed in a shallow inland sea, what's known as an empiric sea, a bit like the Mediterranean. So using that trilobite, we were able to name a new species. So this species we called Lycophoron titan, which is sort of in the center right of the image that you see there. And there's a nice reconstruction of it made by Adam Yates there at the top. These trilobites are a bit like a woodlouse. They look a bit like the roly-polies that are in your garden, except this one, for example, could reach up to 45.3 centimeters long. So it's huge by comparison. Your average trilobite is generally about 7 centimeters long, can fit in the palm of your hand. We found that because it was so big, it actually was the third largest trilobite in the world. You can see the second and first largest trilobite in the world on the left-hand side there of the image. And you can see it's not far off their size in terms of overall body length. Now, being so large as well made me wonder why these organisms became extinct. In particular, these large trilobites seem to have disappeared midway through what's known as the Ordovician period. So what I did was I charted the appearance of the genera, so these are just the different types of trilobites that you have, over time, so moving from the left-hand side of the image is the oldest, the right-hand, which is the youngest. And what we can see there is a group called the acivids, which are these large ones, are part of another group called the Ibexian fauna. This is just basically primitive trilobites. You can see over time, over the Ordovician, the primitive trilobites become extinct or disappear, and the more advanced trilobites, what's known as the White Rockian faunas, seem to increase. And there seems to have been a particular point during the middle of the Ordovician where these large trilobites seem to have done really well as they were crossing over. But why was this the case? Well, again, it has to do with change. If we look at some of the shallow water deposits, so rocks that are formed in shallower waters at the time in Australia, we see an interesting pattern. If we go to Central Australia or far western New South Wales in shallow rocks, we see these acivid trilobites in middle Ordovician rocks, but then those are capped over by Silurian rocks. This is the preceding period. We're missing the latter part of the Ordovician period. However, if we go to deep rocks, like in sort of central New South Wales or eastern New South Wales, we find that the middle Ordovician rocks are capped over by late Ordovician rocks. So what's going on here? Why are these different rocks in different places changing, and why do we have these changes of trilobites? Well, it seems to have something to do with changes in the landscape at the time. Unlike in the previous two examples where I was talking about climatic change, this seems to have to do with geological, geographic change. Australia is formed out of 3 very old continents that are fused together, and the important ones in this case are the northern and southern parts of Australia, which actually form 2 separate continents that are fused together. During this period in the Ordovician, during the middle of it and into the late Ordovician, these two continents started to push really hard against each other. And if anyone remembers back to their Year 9 geology, they would remember that two continental plates colliding force up mountain ranges. And this is exactly what we've got in the centre of Australia, old mountain ranges, which would have been forming at the time, what are known as the McDonald's and the Petermann ranges. And likely as they were forming and going higher and higher, they were pushing the sea further and further towards the east coast. That meant that these aciphid trilobites, which were living in nice shallow waters where they seemed to do really well, were being forced to have less and less space and were being forced out into deeper and deeper waters, where they had to compete with trilobites that had adapted and lived in these deeper waters. That had meant the acipids effectively had less and less space, along with all these other primitive trilobites that love these shallow waters, and the deeper waters had the more advanced faunas in it, and they seem to have done really well. So in this case, we see that geology is restricting the range of these organisms and forcing the change. Now, of course, when I talk about change over time, I'm talking about slow changes over time. I'm talking about things like plates colliding and forcing up mountains, or changes in climate caused by things like the rotation of the Earth slightly tilting or movement of continents over the Earth's surface. But if we think about modern change, this is happening much more rapidly. We see here, for example, things like the Tasmanian tiger as well as the paradise parrot. Both of these are organisms that have gone extinct due to human-induced change. And if we look at the rate of human-induced change, we can see that it far outpaces anything in the history of life. And that's the importance of understanding paleontology. If we don't learn the lessons of the past, how are we going to learn the lessons of the future when these organisms, like the ones that we've already driven extinct, as we start driving more and more species extinct due to things like climate change? So there's a bunch of people I'd like to thank as part of the research that we've done here at the museum. In particular, I'd like to thank the Australian Museum Foundation, who funded the research on those large trilobites and enabled them to be named, as well as understand what was going on in the geology of Australia, as well as Robert Etheridge and Robert Etheridge Jr.'s descendants, who funded the McGraths Flat work and continue to fund it into the future. And a special thanks to all the co-authors of the publications that have been mentioned, as well as all those that have helped us with the McGraths Flat project, particularly the field work, of which there is quite a large number of people that have assisted us in excavation of the site. So thanks very much. I think we'll be moving to a time of questions now.
Thank you so much, both to Professor Chris Helgen for that fantastic introduction about the AM and AMRI, and to Dr. Pat Smith for your fantastic presentation as well. It's wonderful to see all of the fieldwork taking place across Australia and the discoveries and the research taking place at the AM as well. So yes, you're right, Pat. We are now moving on to our Q&A section of the presentation. So please, everyone, we're opening it up with some questions, so please use the Q&A function. And I think I'll just get us started. We've spoken about climactic and geological change over time, human-induced change, and the fantastic terrestrial and aquatic organisms that have been preserved at these numerous sites. So I might throw this to Chris first, actually. So in today's world, what is the benefit of studying paleontology?
Thanks, Megan. And by the way, thank you, Pat, for the tremendous presentation. Great question. You know, what in today's world, why should we study paleontology? I think it boils down to as simple as one word, and that is perspective. Perspective. And so, you know, Pat said it brilliantly when he said, you know, if we don't understand the lessons of the past, you know, how can we be ready for what the future is going to bring? Very true. And this is kind of the core of wisdom itself, you know, understanding what are the interrelationships between things, where do things come from, how have things been in the past, and, you know, how do we use that to shape our understanding of our present world and where we're going. So I think those are huge, big questions that we can derive some really important understanding of from paleontology. And think about the big questions that humans ask about the world. You know, where did life come from? What are the origins of life? You know, who are we as humans, as a species? You know, how is humanity born? You know, where do we come from? The fossil record as we know, as everyone I think knows well, gives us this unparalleled chance to track through time, through real hard objects, fossils in rocks that can be dated and can be studied, to really understand that change through time. So part of it's about where we've come from and where we're going. And then, the other thing, and I'm sure Pat will give us more insight into this, but, we, of course, are in a time of very rapidly changing environments and climates, as Pat pointed out at the end of that presentation. The world is changing, and it's changing very fast. And a misunderstanding of paleontology can often be to say, well, the world has changed a lot in the past. We've had all kinds of different climates. We've had lots of things become extinct. So what's different now? Why should we be so concerned now? Climates have always changed and species have always become extinct. But in reality, the fossil record actually gives us a measurement and an understanding of what we can expect, how much of that has happened through time, the pace of it, and know that where we are now in human history, we're on a different trajectory.
Yeah, I agree. Fantastic. And it is so much about perspective. And you see now with the conversations around climate change, these conversations around, what has happened before, these big climactic events. And I also wanted to hear from Pat a little bit about that as well. So the benefit of studying paleontology, but we also have had a question that links to people studying paleontology now. So it would be great to also hear why you decided to study paleontology and also advice to those who are currently studying paleontology. We have someone currently doing a degree at UNE in science and paleontology, and they're wanting to have any advice about those kinds of studies, what kind of projects look at, and what to do after you've also just studied at university.
I see that person studying it is also named Patrick.
Yeah.
Yes, he's actually the fellow that I mentioned in the talk.
Fantastic.
So this would be Patrick Nelson. So those I studied paleontology because as a child I got fascinated. I had a high school teacher, a father who studied science, and he was fundamental really in my desire to want to understand science better. But particularly, he brought home a video about paleontology when I was a child, and that really got me interested in the thought because it talked about paleontology in Australia, not just paleontology globally. We often hear about dinosaurs in the US, for example, or hear about megafauna in Europe, but we don't hear about fossils in Australia. And really, it fascinated me that this change that happens over time happened in my own backyard, happened to where I was living. And so it made me want to understand the changes that have happened over time in paleontology in Australia, and particularly within sort of the Sydney and sort of east coast as well as central parts of Australia, and even into Western Australia too. What we see is that as the paleontology students who come through, if you're very interested in the subject, you'll want to get involved in things like your local museums, because really they're the central hubs where things like specimens come through, and it's where things like, for example, digs will be organised through as well. And so if you want to get involved in any of this, it's the place to be really if you want to work on paleontology. The advice outside of that is that depending on which areas of paleontology, you may want to do postgraduate work, for example, a master's or a PhD at the university, and that will give you the specialized training needed to understand some of the work that goes on in paleontology. A lot of it's highly technical work, and it requires a lot of detail to understand these things and a lot of training, but it is a very rewarding job. I find it quite rewarding. And so I'm sure my wife would agree with that. And so what you can find with a lot of these students who are interested in paleontology is get involved in paleontology communities, museums, universities, volunteering there, or even things like coming along and visiting conferences.
Yeah, fantastic. Thank you so much. We've had some more questions that are quite technical in nature, and I just wanted to remind everyone as well, feel free to also contact us for the more technical questions and for more detail, but I might throw this one out to you both as well. So why was the first Miocene site, with the fish and mudgies so well preserved?
Well, that has to do with the type of rock it's preserved in. So it's preserved in what's called a ferrocrete. Ferrocrates are generally rocks that actually we don't associate with fossils. But in this circumstance, what seems to have happened is it was a drying lake, possibly caused by things like seasonal change, or possibly in this case caused even by changes in the chemistry. It may not have been drying, but may have had freshwater influxes occasionally. And this has changed the chemistry of the surrounding waters, which were rich in iron. They're rich in iron because I didn't go into this, but there's a surrounding basalt that's actually much older than the rocks themselves. And this surrounding basalt picked up a lot of this iron and moved it into this environment. And the iron precipitated out grain by grain and effectively preserved everything in near complete detail down to the cellular level because it was precipitating around it. This precipitation was probably bacterially mediated, so it had bacteria helping it along, and that enabled it to be so fine and enabled these organisms to be preserved in such fine detail.
I had a chance to go out to see this site with our paleontologists, including Pat, and it's just a tremendous experience. Those fossils are absolutely exquisite, and there's so many of them out there, Pat. Too, I was amazed that you see these absolutely incredible fossils, so many insects and plants and a variety of other things, fishes. But almost every time we cracked open a handful of rocks, there was something to look at, a fossil that emerged. So what a tremendous sight. And that's right, those are just beautifully preserved.
Yeah, and I think it's just fantastic to be able to share those discoveries with as many people as possible, and to kind of highlight again what we were talking about, the benefits of paleontology. And I wanted to, this is more of a general question. I know that the Australian Museum, we do conduct quite a lot of field work, and volunteers are very essential to the museum. We absolutely love our volunteers. And we've got a question about, does the AM conduct field work that people can volunteer for? And that includes in other departments. So I thought I'd throw that over to Professor Chris Helgen.
Sure, thanks, Megan. A really good question. We are really reliant in many ways in the Research Institute on the contributions of volunteers. And those take so many forms. I think that, Pat raised some great examples of, alerting us to sites. One of the ways that we find out about, things that are really important to go out and study is by people raising their hand and saying, this is a tremendous possibility. Would you like to come and see this? And then when we have these big projects underway, there's a need for a lot of hands on deck. And I will let, Pat speak to that, how volunteers have helped at some of these sites. But the answer is yes. We do love our volunteers. They help us in so many different ways on all kinds of projects, including in fieldwork. So thank you for raising that question. We've of course been having the challenges of COVID lockdown, and that means that some of the volunteer work has been on hold, but it also means that, and I think you've seen this well, our science nevertheless has not been on hold in any way. And so this work you're hearing about from Pat, whether it's in the field at times or back at the museum, we're able to keep it going strong. Pat, do you want to say anything about any of the projects mentioned and the way that volunteers have been involved?
Sure, yes. So we've had lots of volunteers involved in some of our projects. So you can see the large list of names that we've had there at the end with some of the people that have visited this McGraths Flat site with us. We've had large numbers of volunteers come along with that. The other projects, for example, the Ordovician site one as well, I've had numbers of volunteers help me with those. So those can come from different areas. The museum itself, we do have some that volunteer through the museum, but we also have groups, the community groups that we've worked with. So, for example, the Fossil Club of Australia, which I've worked with on numerous occasions to help collect fossils from particular sites. In terms of getting involved with those, it's best to generally talk to the AMRI email address, and that will be sent to our curator, and he'll be able to assist with sort of working out where would be best if people want to volunteer and where would be best to volunteer. But we also have volunteers work with us behind the scenes. Of course, what I don't mention as part of this talk, it's nice to talk about the exciting field work that we do, but there's actually large amounts of work involved in cataloguing these specimens and understanding exactly where each one of them comes from. And that requires large teams of volunteers as well. And we need these people to help assist us in these sorts of efforts.
Exactly. I mean, looking at our citizen science projects as well have been fundamental in so many of our discoveries and understanding the data-rich nature of a lot of our projects as well. So I'll move on with some of our other questions now. So I quite like this. It's quite quirky, but been asked if you could be a fossil on the wall, which time period would you like to go back to?
Oh, that's a hard one to answer. I think the Cambrian or the Ordovician for me, just because it's such an alien world. You're talking about a world before plants existed on land. You're talking about a world that's dominated by invertebrates, animals without backbones. A world where CO2 was about as high as it was oxygen. Is a strange alien world, really, for us to think about.
Mm.
I'd I knew that Pat was going to say the Cambrian or the Ordovician because of his love for the trilobites. But I guess if I was to answer that, it'd be much more recent in time. I always dream about what it would be like to have been one of the first people that ever arrived in Australia, setting foot on the continent and seeing the megafauna, the really gigantic marsupials and birds and reptiles that were living on the continent 60, 50,000 years ago. And it's paleontology, teaches us from the very earliest all the way to, the time periods that have just disappeared. And so I would love to go back and see what Australia was like at that moment.
Yeah, it'd be a fascinating thing to see. I mean, I just love Jurassic Park, so I'm going to say the Jurassic period, but I'm obviously a very big amateur as well. So gotta love that pop culture with the dinosaurs. But also, one of our associates has also mentioned that the citizen science project Data Fossil has always been has made quite a big impact as well. And for sure, we would love for everyone to go and explore that on our website. Really important project. I also wanted to pick up something here that's been asked about these fossil discoveries. So, with some of the fossil discoveries, how far have these rocks moved from when they were formed in the location that they were found? So, kind of those, do fossils move very far? Do you have a good way of tracking that?
Well, so fossils don't move per se. What's happened, and there are some situations where they do, but it's a little bit complicated. But most of the time, what you're talking about, when I'm talking about, for example, Australia being near Antarctica and these
Mm.
fossils being moved along, they're being moved along with the continent. The continent has moved as well. And that's because of what's called plate tectonics. We've understood well since the 50s that the continents aren't stable where they actually move over time. And that's just to do with the fact that the Earth's interior moves as well. And as that interior moves, it causes churnings in the different layers of Earth above it. The magma that you get, for example, as well as what we call the mantle. And that pulls along the rock at the surface with it. And so what you've got here is the rocks of the surface, particularly those of the continent of Australia, have been moving continually. Even, for example, before the formation of things like the supercontinent Pangaea, people know of Gondwana or Pangaea when they think about sort of supercontinents over time, of large numbers of continents colliding. But even before that, there was actually supercontinents before that, as the continents broke apart and were moving around. The way we track them is through a complex method of magnetic readings. So you can use magnetic readings in particular rocks to identify exactly where particular continents were at particular times. It gets a little bit trickier as you move further and further back. But sometimes also we use the fossils themselves. So sometimes we find unusual combinations of fossils on continents that you wouldn't expect. And so presumably the two continents were close together at one point. For example, Australia and South China, although they're relatively close today, if we travel further back, they're actually quite far apart. But if we travel further back again, they seem to have been actually closer together at one stage as well. So they seem to have moved further apart and then drifted away and then further moved back together again. It's quite a complex method of different forms of evidence that we use to understand how continents have changed.
That's fantastic. Thank you so much. We are nearly at time. I think we can quickly answer just one more question. So, um, is there one particular fossil that dominates Australia's fossil record? As we've been talking so much about Australian paleontology, I'd love to hear both your thoughts.
I think I'll let Chris go.
I'll send that to Pat first.
Oh. It's a very difficult question to answer. I mean, I probably would end up picking a trilobite, surely.
Yep.
Trilobites are quite common across the entirety of Australia, and I suppose one of the ones that I think is emblematic of the entire country is one called Zistrodura, which is found so widely across Australia. You find it all the way up in the Arafura area near sort of the tropics and into the Cape region, but you find it all the way down here in New South Wales, as well as even potentially slightly into South Australia as well.
That's fantastic. I mean, Chris, do you have a favourite fossil at all that you wanted to talk to at all? It doesn't have to be Australian.
Oh no, I sure do, especially in this part of the world. My background is as a mammologist, and when I think about Australia's wonderful mammals, the monotremes are always at the top of the list. So Australia's egg layers, the platypus and echidna we have today, but that's a really interesting, scattered, but and somewhat slender, but a fossil record that extends back about 125 million years. And so Australia is really the only place, except for one site in South America, where we can watch the evolution of that incredible group of mammals. The other way to be a mammal, that'd be an egg-laying mammal, and that we trace that only in Australia. So I love the monotremes, and their fossil record is absolutely fascinating.
Yep, gotta love those monotremes. Very lucky in Australia to have so much information on our beautiful monotremes as well. So that does take us to time. So I wanted to thank the wonderful Professor Christopher Helgen and the fantastic Dr. Pat Smith for your presentations today and for the Q&A. Fascinating answers. I know that we learned a lot, and it's so great to hear what the Australian Museum is currently doing in the field of paleontology. So thank you again, and thank you to all of our attendees for tuning in. So this webinar has been recorded, and it's going to be uploaded to our AM website on the exclusive online events page. So please keep an eye out for that if you'd like to watch this again and share it with friends and family. So if you have any further questions as well, I know we couldn't get to everyone's today, and some of them were very specific. So please email amri@australian.museum, where a suitable staff member will be able to answer those for you, and we'll be able to respond to those. So take care, everyone. I know it's a very challenging time, but we look forward to seeing you on site at the Australian Museum soon. Goodbye, everyone.